everybody, welcome to the Learning Flock. Today we're going to be talking about food and the nutrients it provides. More specifically, we're talking about biological macromolecules, which are necessary for life on Earth. When you're biting into an apple or cooking with olive oil or even flexing your muscles, though you may not realize it, you're interacting with biological macromolecules. The elements most commonly found in these large molecules are sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen, sometimes abbreviated as sponge. Depending on the quantity and arrangement of these atoms, we get 4 different types of biological macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Each type comes with unique properties and functions, everything from energy storage to genetic information. Well, don't leave me hanging. I want to know more. Well, hold on now. First, let's talk about the synthesis of biological macromolecules. Most of these large molecules are made from smaller, subunit molecules, which are called monomers. Think of them as the building blocks. Monomers link together with covalent bonds to create polymers via dehydration synthesis, where a single water molecule is removed, binding the building blocks together. Dehydration synthesis means to put together while losing water. On the flip side, polymers are broken down into monomers via hydrolysis, where a single water molecule is added. One monomer gets a hydrogen atom, and the other monomer gets a hydroxyl group. Hydrolysis means to split with water. Wait. So when I eat an apple or something, my body is digesting the biological macromolecules through hydrolysis? And when my body goes to build biological macromolecules from those nutrients, it uses dehydration synthesis? Exactly. Now that we know how biological macromolecules are made, let's get into it. We'll start with carbohydrates, a major energy provider for cells that also doubles as structural material for plants, fungi, and insects. They're made up of hydrogen, carbon, and oxygen, often represented by the stoichiometric formula CH2ON, where N is the number of carbon atoms in the molecule. The name carbohydrate literally means carbon, as in carbo, and water, as in hydrate. Carbohydrate? I never knew that. Carbohydrates are classified as either monosaccharides, disaccharides, or polysaccharides. Monosaccharides are simple sugars, like glucose, which has the formula C6H12O6. These are the fuel that your cells run on. Link 2 monosaccharides together via dehydration synthesis, and you have a disaccharide. Sucrose, table sugar, and lactose, milk sugar, are 2 common examples. And get this, the prefix saccharide means sugar. Here's the fun part. If we link many monosaccharides together, more than 2, we get a polysaccharide. Let's review some examples. Glycogen stores glucose in animals, and starch stores glucose in plants. Think potatoes and bread. Both are examples of polysaccharides being used for energy storage. Polysaccharides are also used in structural applications like cellulose, which makes up the cell walls of plants. Another cool example of structural polysaccharides is chitin, which forms the exoskeletons of arthropods. So wait, monosaccharides and polysaccharides are just monomers and polymers, but for carbohydrates? Exactly. This will be a recurring theme throughout today's lesson. As biological macromolecules, carbohydrates are made up of individual subunit monomers, linked together via dehydration synthesis to create polymers. In the case of carbohydrates, the covalent bonds joining monosaccharides to more monosaccharides or other molecules are called glycosidic bonds. Okay, now let's talk about proteins, an extremely diverse and abundant biological macromolecule with seemingly endless applications. Just like carbohydrates, proteins are made up of monomers linked together via dehydration synthesis to create polymers. In the case of proteins, the individual subunits are called amino acids. The covalent bonds created through dehydration synthesis are called peptide bonds, and the polymers are called polypeptides. Amino acids are really quite fascinating. Each monomer has a central carbon atom, called the alpha carbon, bonded to an amino group, a carboxyl group, and a singular hydrogen atom. There are 20 common amino acids, each represented by a 3-letter acronym. I guess that's pretty easy to remember. Hold on now. Let's not forget about that R group, the key differentiator between amino acids. The R group, or side chain, determines whether the amino acid is polar, basic, acidic, or nonpolar. Because each R group is unique, the sequence of amino acids in a polypeptide chain determines the function and shape of the protein. There are actually 4 levels to protein structure: primary, secondary, tertiary, and quaternary. Just when you think it's going to be easy. The primary structure of a protein is the unique sequence of amino acids that the polypeptide chain is composed of. The hormone insulin, a protein, consists of 2 polypeptide chains. The amino acid cysteine's R group sulfhydryl forms disulfide bonds, connecting and shaping the pair of polypeptides. Note that the sequence of amino acids in chains A and B are unique to insulin. The secondary structure of a protein is the local folding of specific regions. Most commonly, we'll see the alpha helix or the beta pleated sheet, which are held together via hydrogen bonds between the peptide backbone. The tertiary structure of a protein, formed by chemical reactions between R groups, is the three-dimensional structure. Ionic bonds, disulfide bonds, hydrogen bonds, and hydrophobic interactions all influence the 3D structure. Okay, how exactly can proteins get more complex? Well, some proteins are so complex that they're formed from multiple polypeptide chains, where the polypeptide chains become the subunits. This is called the quaternary structure. Ultimately, every protein has its own unique sequence of amino acids and shape, which is what enables proteins to carry out such a wide variety of tasks in living organisms. Proteins can be enzymes for catalyzing chemical reactions, they can be hormones for chemical signaling, they can serve as structural materials or contractile fibers. Even antibodies are proteins, an integral part of your immune system. Holy guacamole, these guys are talented. Next up, nucleic acids, the macromolecules that are responsible for the storage and transmission of hereditary information. With nucleic acids, the individual subunits are called nucleotides, the covalent bonds are called phosphodiester linkages, and the polymers are called polynucleotides. There are two nucleic acids to remember: DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. Now, every nucleotide has three parts: one pentose sugar, deoxyribose or ribose, one phosphate group, and of course, one nitrogenous base. There are 5 nitrogenous bases found in nucleic acids: adenine, guanine, cytosine, thymine, and uracil. Cytosine, thymine, and uracil are pyrimidines. They have a single carbon-nitrogen ring structure. Adenine and guanine are purines, which are slightly bigger with their double carbon-nitrogen ring structure. Well, that's kind of cool. It's almost like nitrogenous bases are some sort of alphabet. Exactly. Let's talk briefly about the difference between DNA and RNA. DNA is the genetic material of all living organisms, from single-celled bacteria to ducks, pigeons, and humans. And while DNA stores the instructions, different types of RNA carry them out. mRNA carries the message, rRNA forms the ribosomes, tRNA delivers amino acids, and microRNAs regulate gene expression. We can't forget DNA's classic structure, the double helix, where 2 anti-parallel strands twist together while running in opposite directions. On the outside, the pentose sugar and phosphate of each nucleotide form the backbone of DNA. But the real fun happens on the inside, where the nitrogenous bases pair up. But only certain purines can pair with certain pyrimidines. Adenine pairs with thymine, and guanine pairs with cytosine, sometimes called the base complementary rule. That means that if one strand of DNA is GGTTCCAA, the complementary strand would be CCAAGGTT. On the flip side, RNA is usually single-stranded. Additionally, keep in mind that a DNA sequence of GGTTCCAA will result in a complementary mRNA sequence of CCAAGGUU because DNA gets thymine and RNA gets uracil. This flow of information from DNA to RNA to protein is called the central dogma of life. Now, last but certainly not least, let's talk about lipids. They play a major role in energy storage, insulation, cell membranes, and even chemical signaling. Just like carbohydrates, lipids are mainly composed of hydrogen, carbon, and oxygen, but they're arranged differently. Lipid superpowers that they're usually nonpolar and hydrophobic, which means water-fearing, making them insoluble in water. So there's superpowers that they're scared of water? Okay. Unlike the other biological macromolecules, lipids are not true polymers because they aren't built from long chains of repeating monomers. You'll see in a second. There are several major types of lipids to remember: fats and oils, phospholipids, waxes, and steroids. Fats are made up of 2 key components: glycerol and fatty acids. 3 fatty acids bond to glycerol's 3 carbons via ester bonds, releasing 3 water molecules in a dehydration reaction. Because of the 3 fatty acids, fats are sometimes called triacylglycerols or triglycerides. Additionally, fatty acids can be saturated or unsaturated. If a fatty acid is saturated, that means that we only have single bonds in the hydrocarbon skeleton. If a fatty acid is unsaturated, that means that we have at least one double bond between carbon atoms, which leads to bends in the chain. A great example of the difference would be olive oil, mostly unsaturated, and butter, mostly saturated. Well, I guess I ate too many saturated lipids over the holidays. Let's review some other types of lipids. Waxes are hydrophobic and act as natural water repellents. In nature, they sometimes coat the feathers of aquatic birds and the surfaces of plant leaves. Next, we have steroids, which are quite different from other lipids. They have a fused ring structure, but are still grouped together with lipids because they're hydrophobic and insoluble in water. Cholesterol is a very common steroid. Despite its bad reputation, it's really important. Your body uses cholesterol to produce hormones like testosterone and estradiol, and it's also a precursor to vitamin D. So not like gym steroids, but the important ones. Exactly. Lastly, we have phospholipids. These are supremely important because they are the primary components of cell membranes, the phospholipid bilayer. Now, unlike fats, phospholipids have just two fatty acid tails, forming diacylglycerol with a modified phosphate group occupying the final carbon of the glycerol backbone. The neat thing about phospholipids is that they're amphipathic, which means that they are partially hydrophobic because of the fatty acid tails and partially hydrophilic because of the phosphate. One end loves water and the other hates it, making phospholipid bilayers possible. As you can see, biological macromolecules are really super cool. They turn the food we eat into energy, structure, signals, and even the instructions that make us who we are. And that wraps up our overview of biological macromolecules. If you liked today's video, make sure to give it a like and consider subscribing for more or following us on Instagram. Drop a comment below and let me know what kind of topic you'd like to see next. Thanks for watching The Learning Flock, where your grades take flight.